Hello.
Yes, sir.
Okay. So some of the things we'll quickly review, which we have seen earlier. Uh, Human body contains around 5 into 10 raised to 13 cells, and these cells can be of different categories. That is, they can be non-dividing and terminally differentiated. Example, bone is not dividing and it is terminally differentiated, or even neurons. Those, there can be some of the cells are continuously proliferating, means they are growing, they are increasing their size. They may be in rest, but they may be recruited into cell cycle. We know phases, if you remember, G0, G1 phase, and so these are various cells. And tumor becomes detectable clinically when there is a mass of 10 raised to 9 cell. It becomes around, that is called as a 1G. Okay. Now let us see what is the cell cycle, which we have seen earlier. We will just repeating. Okay. Normally cell is a resting cell is in a G0 phase. It has got before that it goes to G1 phase. From G1 it goes to synthesis phase, G2 phase, and then mitosis. And once mitosis is there, this G0 phase is there. That is, we can say one is nascent or is not doing anything, and another is the G0 which actively gets converted to G1, like that. So this is what we have seen. Once there is a mitosis, then there can be either cell division or there can be cell differentiation, and later whenever the time comes, the cell can get its own death naturally. So this is what cell cycle we have seen. The important thing is cell differentiation, cell division, and cell death. These are very, very important features in a cell division. We have seen already this, so there is no need to go for more details. That is, there is a growth occurs, and what happens during G1, S phase, G2 phase, M phase, we have seen when we were studying about cell. So this is how the cells are there. Now let us see what is the difference which is taking place, because when it occurs normally, then we get normal cells where the oncogene, you know now oncogene, because this also we have studied. Oncogene expression is rare. Intermittent or coordinated growth factors, they are secreted, and they are coordinated when they are not too much excess, like that. Presence of tumor suppressor genes are there, which are controlling the cell division or cell differentiation, and very few mitoses are there into it. When we compare it with the cancer, because normal cell we know about, but when we compare it with the cancer cells, we will find that there are very frequent mitoses, that is frequent this mitosis activities will be there into it. Then you can see the changes in the nucleuses will be they will be changed in the size, because mitosis again will be going into that. Then loss of contact inhibition is there. Increase in the growth factor secretion will be there. Because of increase in the growth factor, more cell division chances are there. There is an increase in the oncogene expression, and there is a decrease in the tumor suppression gene which are available. So these are various contributing factors which are contributing for the cancer development. So there are growth factors, as we have seen. There are so many growth factors which are acting on cells, and they are stimulating the cell divisions. If you remember the blood formation, in that we know various growth hormone growth factors. Then EDRFs we know, that is endothelial derived growth factors, EDGF vascular growth factors. There are so many growth factors, many growth factors are there. So these all are growth factors. They can be acting autocrine way or paracrine way. Autocrine means on the same cell, paracrine means adjacent or nearby cells. They will be acting. Because of that, they will cause the post receptor signal transduction pathways will be activated. Because of that, there will be gene activation, and oncogenes will be activated. Because of that, more growth factors will be released, and more receptor synthesis will be there. and these more receptors and more growth factors will be causing more cells to duplicate or differentiate, and this would result into more problem. So why there is a cancer? Cancer is because of the various things. If we compare it, let us see what happens. So this is the normal process which is there, that is cellular material is there. So it can be either viral oncogenes. If you remember, anybody remembers, recently we have seen which viruses are responsible for cancers. Yes, viruses that are responsible for cancers.
Sir, Epstein-Barr,
Epstein-Barr is one. Okay, then
um, papilloma virus,
papilloma is there, and cytomegalo, most important is cytomegalovirus, CMVs. So these are the viruses, they carry oncogenes, they insert into the human genetic sequence, and then they will be responsible, or they will be doing. And there are some agents which are carcinogenic in nature, or they will cause mutagenesis, and they will thus activate the cellular oncogenes. Because of that, there is increase of the abnormal expression, and that will result into the cancer. Okay, if we have normal oncosuppression gene is there in working, then there is a normal expression, and because of this normal oncosuppression gene is there which is working, then there will be normal growth and development. So that balance between oncosuppression, okay, and oncogene, that is very very important. When this balance is lost, then there are chances of cancer. So again we have seen CDKs play very very important role in cell cycle. If you again go back, or you just revise it again, and this CDKs they play very very important cyclin dependent kinases, and they play a role at various places. There are a variety of these agents, and these agents are playing very very important role in cell cycle. If you remember, they are also important checkpoints. You just revise it what we have studied earlier. So these are important checkpoints, and they play very very important role uh, in activities, that is cell division, cell differentiation, checkpoint activities. So what are these cyclins? Are they no intrinsic enzymatic activity? They binds the cyclin dependent kinases, and these are synthesized and degraded in each cycle. The essential components for CDK activity are the cyclins. So when cyclins are there, then only CDKs can do anything. So cyclin CDKs, they are regulated by tyrosine 15 phosphorylation inhibitory kinases. Then they are activating phosphatases. They causes direct interactions, and there are very very important proteins are there which are playing either excitatory or inhibitory activities. Example of these are P21, P27, P57. Again, these proteins also we have seen. P57 plays very very important role in cancer activities or apoptosis activities. P16, P15, P18, P19, these are various proteins. They are produced by these activities through cyclin and cyclin dependent kinases. So there are cell cycle checkpoints again that we have studied earlier during cell division and all. So everywhere these checkpoints are there. We have seen that if the checkpoints are clear, checkpoints is very very rigorous check made by this checkpoint system, and if anything goes wrong, then they will not allow this cell division or cell differentiation to complete, and that will be terminated. And but because of one or other reason, if these cell cycles they are not working properly, if they fail to do it, then we can have chances of having oncogenes or undifferentiated or poorly differentiated or multiple growth cells into this. So there are variations are there in the cell cycling. These CDKs are having varieties, variations into that in different phases. If you see uh, this CDK, that is cyclin dependence kinase 4 to 1, they are playing role at multiple places as and at that is from, you can see that is from mitosis G1 phase, S phase, G2 phase, again mitosis, again G1. So this is where the cell division starts, and at that place these are the various subunits of CDKs, they are acting on various places. So they are very very important as we have seen earlier, and they are playing very very important role at multiple places. So again, when we go for cell cycle, we can see that what happens, and the duration if we see, it looks complicated, but these are these various cyclin E and cyclin CDKs, they are playing role into G1 to S phase. We'll start from this. There are cyclin Ds and cyclin 4 cyclin Cs, they are from G0 to, they will bring to this G1 phase. From this G1 phase, as we have seen, there are even they are playing role in this also, that is G1 to S phase. Then from S phase again cyclin A and CDK. So these are various CDKs. They are playing important role at vertip because they are checkpoint at multiple places, we know, and there are multiple type of CDKs. So they are playing important role, and they are very very important in the normal cell division. There are variety of proteins are there, example P53, P21, they are having various mechanisms, and they are responsible for various actions, including the arrest of cell division and the inhibition of that particular activities. So these are P53, P57, P21, they are most important one, which are responsible for that, and they are rather we can say anti-proliferating type of activities. DNA damage are there, they are made by this, and they can thus once DNA is damaged, they can arrest the cell cycle, and thus we call that as a damage dependent checkpoints are there, and these checkpoints again are important, as we have seen. If this earlier, that if this checkpoint shows that there is a damage, they are not proper thing, then they will not allow to things happen. Then next is G1 to S checkpoints, and we can see that because of X-rays or some other things, the strands will be broken, and that will result into formation of P53, P21, and these agents, that is cyclin dependent kinase inhibitors are there, we call them CKIs. So it will act as a CKI inhibitor, and therefore it will inhibit the cell division. N terminal part of 21 forms complex with cyclin CDK inhibition kinase, and thus it will inhibit. Cell cycle is regulated through such various genetic activities, and these things, they will be, example, there is a DNA damage which will activate P53, and that will through P21 it will inhibit this CDK complex, and thus it will affect the further activities. Then growth factors are there, as I told you, there are many multiple growth factors are there. These growth factors act through their receptors, either cellular receptor, nuclear receptors, and thus they will be by any mechanism they will be causing the initiation of cell division, and therefore this will be developed. Then next is, this is just basis we are going for, so that we will understand some of the basic important thing. Then there are some other important proteins like retinoblastoma protein, that is called as PRB, and CDK inhibitors like P21, P27, P16. They are again playing important role in all this cell cycle arrest, that is they will not allow cell cycle to grow, or cell cycle to make duplication, as I told you what it is, that is cell division or cell differentiation, they will not allow. So these are various activities, these are various growth factors which otherwise are doing, but if there is a DNA damage, then this will inhibiting the activities. The normal cell cycle and cyclins of the cell cycle, what they are doing is, this is the normal cell cycle, and where the tumor suppression and apoptosis is active. So whenever it is there, the quiescence, differentiation, then senescence, apoptosis, this all things they can apply, they can happen during G0 phase, and there will be arrest of G1 phase because of one or other activities, maybe tumor suppression and apoptotic genes, they will help in apoptosis, and the cell number, cell count will be in control. But in case of neoplastic cell, they are called as immortals because they are not dying, because their apoptotic activity, the apoptotic activity, that is the cell death which should have been occur in given time, that should, example RBCs, we say RBCs has got lifespan of 120 days. After that RBCs should die, they should be taken up by liver and they should be metabolized to heme and globin, that you might have studied. So that is the life cycle of RBC. That means after 120 days or in this menstrual and ovarian cycle, you may know that is there is known as a corpus luteum that is formed, and that is destroyed automatically because that is the lifestyle, that is the lifetime of that particular thing. So this is what happening, because these are the mortal, okay. But this becomes, when these growth factors, they are impacting on this cyclin D and cyclin E's, so they will be continuing that, and this E6, E7, these are the proteins, E5, E6, they will causing immortalization of the human keratinocytes. That means they will not die. If they do not die, that means they will continue be formed, and if they are continually formed, they will be causing cancer. So these are various reasons why there are cancers, because why I told you is most of the in book will find all the classical drugs only, but recently when we have studied about cell division and all, we have found many drugs which are acting at newer targets. Example, if you say that imatinib mesylate and all, okay, then there are various monoclonal antibodies which are working at various such cyclin dependent kinases or CDKs, RP53s, etc. So these are the newer analogs. These are going to be very, very important contributors in the treatment in the future. What are the older drugs which are there? They are very clear when they are available. Example of these are alkylating agents, antimetabolites, anti-tumor antibiotics, alkaloids, taxanes. We shall see in detail one by one. We'll see examples and how it is. Now alkylating agents are the drugs which are interacting directly with the cellular DNA. Very nice figure is given in Goodman and Gilman and even some of the medicinal chemistry book, that what happens is these agents, they bind with the there is a base pairs are there, AT and GC, okay, that is adenine, thymidine, guanine and cytosine. So we call it as AT or GC, and this base pairs, when these alkylating agents are there, let me try, or will go for mechanism in more details later. What we'll do is like that, we'll just have a classification, partial classification, few more drug will add. So they will be alkylating it, because alkylating means they will get attached, just like simple, if one methyl group is attached, we call it as alkylation, because methylation or ethylation, this is all nothing but the alkylation. So they will getting attached to that particularly any kind of nucleotide, and because of that they will change the conformation of that particular thing, maybe adenine, guanine, cytosine, thymine, whatsoever it may be, and because of that they will change the base pair sequences, or they may cause ruptures, they may cause damage. We shall see all this. So these are known as alkylating agents. Then antimetabolites, we have studied them in antiviral or antibacterial, all cases. We call them as antimetabolites. Why we call them antimetabolites? Because they will not allow to form the normal metabolite, which is very, very important. May it be these folic acid analogs, that is tetrahydrofolate, whatsoever it may be. So these are the important metabolites which are either required for either protein synthesis or nucleic acid synthesis, DNA, RNA, etc. Wherever these are required, the raw material will be destroyed. This raw material will be resembling it, but it will be useless raw material. So we call them as a antimetabolites. So they will resemble the cellular metabolites like folic acid, purine, pyrimidine, as I told you, and they will interfere with the DNA precursor or cellular metabolism. I repeat, already I told you, antimetabolite doesn't mean, anti means against, metabolite means, that doesn't mean that, or here the word inhibition comes, it doesn't mean that that will be stopping the action. Actually it means that there was a car was meant for some X, okay, a similar car is coming, the X will not able to sit into it, and therefore that car will not go to the destination, okay, but car is going, car is there. So suppose enzyme is a car and the car was supposed to carry X person. Now the other person which is resembling the dress and everything like X, or having identity card like X, will show it. The car will take that Y person who is not of any use. The car will take the Y person, X person will be left behind, and because of that the work which X should have been or X could have done, that will not be done. Therefore we call it as a inhibition, but process is not stopped. Then next is anti-tumor antibiotics. These are the agents which are obtained from variety of sources. So these are maybe from fungus or some other activity, and they are having variety of action. There are variety of these antibiotics. We shall see each of them. Then there are agents which are derived from various plant extract. We call them as a mitotic inhibitors. If you know the basic physiology of cell division, you will find that there is a formation of mitotic spindles and the microtubules and this various tubular structure that play very, very important role into it, and thus these mitotic spindles or mitosis or these microtubules which are playing important role in formation of mitotic spindle. These microtubules, they play role as a, you know, just like a cable car is there, so they are helping like that, just like a cable car or ropeway. This mitotic spindle will allow the cytokinesis. So this will be affected when these mitotic inhibitors are there. So let us see some of the example of that. We have seen basic mechanism also. Alkylating agents, example of these are carboplatin, cyclophosphamide, melphalan, thiopet, and there are many actually. There are alkylating agents are there. I told you they form bonds with the nucleic acids and protein, and therefore they make them inactive or alter their structures. Then antimetabolites, example of these are methotrexate, fluorouracils, gemcitabine. You will find that they are mimicking the structure and name are clear. Methotrexate is antimetabolite for tetrahydrofolate synthase. Fluorouracil is uracil, and gemcitabine is for these various purine synthesis. The natural products are there. There are many natural products which are like anti-tumor antibiotics, microtubule stabilizers, mitotic inhibitors, topoisomerase inhibitors. An example of these are doxorubicin and daunorubicin. One more, like example docetaxel or paclitaxels. These are all taxel compounds. Then vinolbin or vincristine, vinblastine, etc. There are many vinca alkaloids. Then tapotecan, and there are so many. There are endocrine agents, very important condition. This is important is we know that hormones are playing very important role in proliferation of certain tissues. If you remember, we know progesterone, we know estrogen, we know testosterone, how they are playing important role in various proliferation of gonad or gonad related tissues. So if we can inhibit it, then accordingly we can have this. So these are various drugs like anastrozole, tamoxifen, prednisolone, goserelin, which are acting as an aromatase inhibitor. Aromatase is an enzyme which is very important in testosterone metabolism. Then there are estrogen antagonists we can use. We can use corticosteroids. We can use the luteinizing hormone, releasing hormone agonist. There we can use them. So there are multiple such endocrine agents. Particularly, they are very important in the cancers which are based or which are related to the uh, secretion, more or less secretion of one or other kind of hormone, and or the where the tissue growth is depending on the hormone secretion. So in that condition we are when it is linked, example as I told you, gonads and all. So in that cases these endocrine agents are very useful. Then the newer classes are there are very important newer classes. I told you just like that is imatinib mesylate was I don't know, but since last 6 year I am seeing the message every year as a new or very important information. People may not able to spell it properly, uh, spell it anything like that, but will not spell it correctly, imatinib mesylate, and they will say that this and this company or this and this place, mostly either Chennai or Pune, they say that this drug is offered in free of cost to the cancer patient and is very effective drugs. Nobody knows exactly in which cancer in type of cancer it is very effective, and what it means, whether really this free drug supplies they are available or not, but people are forwarding like anything. So these are the drugs, these are the very new drugs. They are, we call them as a molecularly targeted agents. So molecularly targeted, example for that, again we have to go back for our basics which we have started, which we have done it earlier. I told you that there are many mechanisms which are involved, various proteins, various like P53, P57, P21, and many more. Then there are CDKs, then cyclins, and many such things are there. So we have got drugs at multiple level, and accordingly we have got drugs which are acting on gene expression. There are drugs which are acting or which are available as a monoclonal antibody against various growth factors or various factors which are affecting. And important is tyrosine kinase inhibitors. Example is imatinib mesylate or gefitinib. These are the newer drugs which are there. Then there are various drugs which are, we call them as a biological response modifier, like interferons, thalidomide, filgrastim, which are helping in modification of the response. And these all classes, they are helping, they are very important in one or other way. So alkylating mechanism, I'll go from, I'll suggest to read from particularly Goodman and Gilman. I'll try to show it from Goodman and Gilman, just a minute, because that will help you more. So let us go. These are various classes which are available, and we shall see mechanism of each. These are the steps which are taking place, and accordingly what are various drugs which are available, step by step we'll go by. So we know that purines and pyrimidines are responsible or required as a important base material for formation of either RNAs or DNAs. Okay, so these are formed. There are enzymes which are playing important role in formation of these. So these are the purines and pyrimidines which are there. We have got purine analogs like 6-mercaptopurine and 6-thioguanine. They are the agents which are purine analogs. They will inhibit the purine ring biosynthesis. They will inhibit the DNA synthesis because they will compete for the normal purines. Okay, and then the next is this pemetrexate, methotrexate. These are the two drugs. Pemetrexate is a newer analog of methotrexate. Methotrexate is a very older one, and this is acting, these drugs are acting at multiple places because this tetrahydrofolate is very important as a coenzyme. Again, biochemistry we have to study for that. They are very important as a coenzymes, and these coenzymes, they are as a coenzyme, if it is not properly formed, then they will not able to go for this kind of synthesis. Okay, so this is what methotrexate is doing, and thus it will not allow these things to there. So they are inhibiting the dihydrofolate reduction. They will block the thiamidylate and purine synthesis, and thus once it is blocked, then no DNA can be formed. This is very important action they are playing with. Then one more drug is there, that is hydroxyurea, that will inhibit the nucleotide reductase enzyme. Again, it plays very important role in the nucleotide synthesis. Then 5-fluorouracil, that will inhibit the thiamidate synthesis. 5-uracil that gets incorporated in place of thiamidylate or thymidine, and thus it will be there. Then next thing is this drugs are there like gemcitabine, cytarabine, fludarabine, 2-chlorodeoxyadenosine, clofarabine. So these are all analogs which will be getting incorporated into this, and they will form the false or wrong tissue, and thus because of that the DNA synthesis will be inhibited. Now next is, once DNA is there, then the next step is in DNA. Whenever DNA is, if you remember, it is wound with the histones, and we need that unwounding activity. We call them as a topoisomerases. You have to just recall it. So these topoisomerases are very important for unwinding of that. Whenever any replication or transcription has to be done, then topoisomerase will cause the rotation of it, so that this unwinding will take place easily, and only selective portion of the DNA will be available for either transcription or replication. So there are various drugs which are affecting these. These are example of these are camptothecins, etoposide, teniposide, daunorubicin, doxorubicins. Daunorubicin, doxorubicins are the antibiotic, while these are the plant-based compounds, etoposides, teniposides are. So they are blocking the topoisomerase functions. Then we have got platinum analogs, alkylating agents, mitomycin, then temozolomide. These are all are forming adducts with the DNA. Adducts means they may bridge two DNA. Two strands are there. These strands will be covalently bond. If these strands are covalently bond, then what will happen? We know that for any kind of transcription or replication, this DNA has to separate, so that one strand is used as a template. But if this is a covalent bonding, then this will not be allowed, and because of that, this actions, this will not be there. So these are the agents like platinum analogs, cisplatin, carboplatin, etc. Okay, then alkylating agents, there are many, we shall go for them. Mitomycin, temozolomide, they will form adduct. Then next is another enzyme. Now what we have seen, that this topoisomerase are responsible for separation of strands, unwinding, therefore single strand DNA is formed, and that is used for transcription or what we can say replication. But this will not allow, one is topoisomerase, another will form the covalent bond, so single strand will not be formed. Now even if single strand forms, then we have got various agents like L-asparginase. It will cause deamination of the aspargine, and if the aspargine is deaminated, then that aspargine is required in quite more amount to form the protein synthesis, and this protein synthesis will not be taking place, is another thing. Then protein kinase inhibitors and antibodies are there, which will act on various enzymes which are playing very important role in cancer formation. So we have got various monoclonal antibodies and all other things. Then we have got checkpoint inhibitors, which will block the immune evasion of that, and they will block the response for the differentiation to the antigens. We have got hormone antagonists. They are particularly useful in the cancers which are having causative agent of hormones. In that cases, these hormonal agents, as I told you, estrogen, progesterone or testosterone modulators, we can say, they will be useful in that conditions. Then once these proteins and all are formed, then next is important step during cell divisions or differentiation. Particularly cell division is what, that this cell division, this all things are to be there is a cytokinesis, that microtubules will carry it towards the centrosome, and microtubules, they will play important role in cytokinesis and cell division. Mitotic spindle will be formed. So these epothelions, taxanes, vinculclide, then stramastine, they will be playing role in this inhibition of the microtubules, or the mitotic spindle formation will be affected, and because of that cell division will not be possible. And lastly, differentiation is important, that the cells which are grown, they will be differentiated into one of the prominent type of cell. An example of these are atra, arsenic trioxide, histone deacetylase inhibitor. These are the classes of drug which will either induce differentiation, and accordingly they will be acting. So these are various drugs which are this is the classification of anti-cancer drugs, and basically this is mechanism based. In chemistry you will find they are purines, pyrimidines, like that. Here we are classifying on the basis of mechanism, and particularly we have got it in a sequence, that at what stage or stage wise mechanism we have discussed, site of action at different stages and how they will be acting. Okay, then there are agents again, how they will be acting at different places. Example of these are, we know again cell cycle. There is always G0 is a resting one, and it is always having interconnection with the G1 phase. So what are the drugs which are acting on the cell cycle? Non-specific drug. So it doesn't mean that cell cycle should be at any stage of the cell cycle. That means even G0 they will be able to active. Okay, so these are the alkylating agent, nitrosoureas, anti-tumor antibodies, antibiotics, procarbazine, cisplatin, dacarbazine. These are all the drugs, they can be acting at even G0 phase, even though cell is in nascent stage. As it progresses G1 to G2 phase, from G1 to G2 when it goes, then we have got various drugs which are acting on G1 to S phase, that is CDK4 or CDK6 inhibitors, and there are many new drugs are available in that, that is the palbociclib, then abemaciclib, then ribociclib. When it is IB at the end, that means what I told you earlier, I suppose. If it is MAB at the end, we call it as a monoclonal antibody. Okay, if it is IB at the end, then these are the tyrosine kinase inhibited, imatinib. Then just now we have seen this one along with imatinib, what it is. So all these clips are there and nibs are there. Nibs are NIB nibs. Imatinib is what? Imatinib is tyrosine kinase inhibitor. Then there are drugs which are acting on synthesis phase. You know what is synthesis phase? Synthesis phase is a phase where there is a synthesis of various material, including the material for the new nucleic acids, for the new cells. Okay. So these are the cytosine, arabinoside, hydroxyurea, irinotecan, topotecan. These are various drugs. Then S phase specific self-limiting drugs are 6-mercaptopurine, methotrexate, etc. And the drug which will be attacking mitosis, okay, that is mitotic spindle formation. These are vincristine, vinblastine, paclitaxel, and these are many drugs. So we have done it in two ways. We have done two types of mechanism of action. We have done it on the basis of cell cycle base, that is G0, G1, G2, G1S, G2, and this mitosis. So these are two ways by which we can classify, we can see that how these are acting. Okay. Now next is various important what we can say mechanism of actions. One by one we can go for this. So very first one is the alkylating agents. There are many alkylating agents. You can see the name of these. These are first class is called as a nitrogen mustards. In that there are many drugs like meclorethamine, cyclophosphamide, E-phosphamide, melphalan, chlorambucil, then bendamustine. These are all are from the class of that is called as a nitrogen mustard. Another very important class in the alkylating agents is the ethylenamine and methylenamines. Example of these are thiotepa was older one, newer one is altritamine. Then another class is there that is called as a alkyl sulfonates. Example of this is the basulfan. Nitrosoureas are carmustine, streptozotocin. Then triazines, that is dicarbazine, then temozolomide, methylhydrazines, that is procarbazine, platinum compounds, example of cisplatin, carboplatin. Then these are all drugs which are acting or they are coming under class of alkylating agents. Okay, other class we shall be seeing later. So first class is alkylating agent, or rather we will see all the classes first. Then another is antimetabolites, folic acid analogues like methotrexate and all, they are there in this. Pyrimidine analogues, there are many pyrimidine analogues like fluorouracil, fluorouridine, then capecitabine, trifluoroiridine. Cytidine derivatives like cytarabine, or it is also called as a cytosine arabinoside or araci. Then azacitidine, that is 5-azacitidine, D-cytosine, gemcitabine. Then there are many analogues from purine class, that is thiopurine analogues. Simply what happened at 6 level, if you see mechanism this structure activity relationship in medicinal chemistry, you will find that at certain group the structure is changed. The example, normally thio groups are not there, we are inserting thio group, or structural modifications are made in such a way that when they are incorporated in DNA or RNA, it will not be functional. Then fludarabine, cladirabine, clofarabine, nelarabine, pentostatin, these are various purine analogues. There are many natural products, vinca alkaloid, vincristine, vinblastine, then eribulin, taxanes, that is paclitaxel, then other taxanes, uh, istramastine, epothalones, these are various drugs. Then next is camptothecin analogs. Example of these are topotecan, irinotecan. Two important antibiotics are there. As said, doxorubicin is also antibiotics, but we do not call it as a, or we do not take in that way. Antibiotics like dactinomycin or actinomycin D is a antibiotic. Then anthracyclines and anthracycline dienes. Then epipodophylotoxins like etoposide, teniposides, these are from podophylline. Drugs of diverse mechanism of actions, there are many or more than one mechanisms like bleomycin, mitomycin, mitotane, then trabectedin, L-asparginase, hydroxyurea, retinoids, arsenic trioxides, and other hormones. So anti-cancer, you have got a very big range of drugs. Many drugs are there. Some of drugs are selective, some are non-selective. So far what we have seen is the drugs which are acting, which are at various levels. So very big and very huge classification this is having. Now this is how the drugs which are acting for alkylating agents, you will find that they gets activated once they enter into body, and they get incorporated, or they form the bond, and because of this bonding, okay, because of this bonding, now this has got bonding, it has got attached to this group. This was the drug which has got activation. Because of activation, it has got an active site over here, that is N plus site, that is an active site. This active site or this activation, because of this activation, this drug has become very active, very reactive in nature when it comes into contact with this DNA having this purine structure. So on this purine structure it will bind. See, this is purine structure, this one purine, then second purine or pyrimidine, and accordingly there is a chain. Here DNA means they are having attachment. There are two attachment. One is in chain and another is intercalation between other chain. Intercalation is a weak bonding. Okay, so when these drugs are there, they will form the covalent bond, and they will affect these intercalations and this bonding. So these are very important actions these drugs are showing, and because of that these agents will show multiple actions, and because of that alkylations they will cause variety of action. Either they will be able to bind with the enzymes and inactivate the enzyme, they will block with that, they will act because of the covalent bonding, they will not allow this strand separation of DNAs or RNAs, or they will able to change the chemical nature of the compound in such a way that the adenine will be recognized as a guanosine or like that. That means the base pair can be changed, the what we can say complementary bases, because of the change in the structure, the different type of complementary base will be formed. So there are such multiple actions they are showing, and these multiple actions will result into difficulty in the either replications or problems in the replications, and the problems in the what I say in transcriptions. So this is how these alkylating agents are acting. We shall see again the next detail in our next class. This is very big topic, otherwise please simultaneously have reading with this so that it becomes easy for you. Okay, if you have any difficulties you can ask. So there are multiple things are there in this. Please go through this. Now coming to
हॅलो सर.
yes.
हॅलो.
Yes, listening, tell. हॅलो.
झालं का सर तुमचं? मला थेरा प्रॅक्टिकलचं विचारायचंय.
तेच थेराबद्दलच बोलायचं मला. बोला तुमच्या
मतलब
डिफिकल्टी.
आपल्याला केस प्रेझेंटेशन करायचंय
हो
लेक्चर मध्ये,
हो.
तर त्याच्या आधी सर्वांना सगळे सोप आपल्या ग्रुपवर अपलोड करायचे आहे का?
नाही, सगळे अपलोड नाही करायचे. तुम्हाला काय करायचं आहे मी परत रिपीट करतो. व्हॉट यू आर टू सपोज टू डू इज समजा आजपासून आता आपल्याला 8 दिवसाचा वेळ राहिलेला नाही बेटा. आता एव्हरी 3 days यू हॅव टू गो फॉर प्रॅक्टिकल बिकॉज
ओके.
वी हॅव वेस्टेड सो मेनी डेज. Okay, so we'll do 2 days. One is Monday and next will be the Thursday. या 2 दिवशी आपल्याला कंपल्सरीली प्रॅक्टिकल घ्यायचे आहेत.
ओके.
ओके,
मीन्स पर वीक आपल्याला 2 केसेस घ्यायचे आहे ना?
2 केसेस घ्यायचे आहेत कारण ऑलरेडी खूप डिले झाला आहे. नाव हाऊ वी आर गोईंग फॉर दिस.
ओके.
ओके, लेट अस सी द मोड. जो केस आपल्याला सोमवारी प्रेझेंट करायची आहे तिची या सोमवारी तुम्ही घेऊन ठेवा. जी केस तुम्हाला थर्सडेला घ्यायची त्याची या थर्सडेला केस घेऊन ठेवा. उद्या थर्सडे आहे, उद्या तुम्ही त्या थर्सडे करता डे 0 झाला. म्हणजे या उद्याचा जो थर्सडे आहे तो डे 0 आहे. नेक्स्ट थर्सडेला तुम्ही केस प्रेझेंट करणार. आणि याच्या आधीची जी मंडेची होती, व्हॉट यू विल डू नाव, ही केस तुम्ही ग्रुपला टाकणार. यू विल पुट द केस इन ग्रुप. यू कॅन सायमल्टेनियसली टाईप इट डाऊन टू आवर ग्रुप. आहे ना, ग्रुपला तुम्ही आपल्या व्हॉट्सॲप ग्रुपला टाका. आय विल चेक इट व्हॉट युअर वेदर यू आर अंडरस्टँडिंग इट ऑर नॉट. एव्हरीबडी जस्ट पुट. ओके.
येस सर.
So first step is first thing is I'm checking on the group now. Every Thursday and every Monday we should have the case presentation. Okay.
ओके.
Now suppose any
सर
day.
अजून एक
ऐक ना, पूर्ण ऐक. मी पूर्ण एक्सप्लेन करतो, मग तुझ्या डिफिकल्टीज आपण सॉल्व करू.
सॉरी.
Now when we are going for that Monday or Thursday, suppose tomorrow we are starting the case, and when we are starting tomorrow, then tomorrow you are displaying the cases. Simultaneously अजून कोणीच लिहिलं नाही ग्रुपला. I am just observing this. I have asked you to put a sequence of it. So tomorrow on Thursday you are taking the case, you are displaying to everyone in the class. That means in our common group. But what you are doing then, जो तुमचा ग्रुप आहे पर्सनली 4 लोकांचा, then you will have discussion on that and everybody will write its own interpretation of the case, its own SOAP format. ओंकारच्या टीममध्ये कोण कोण आहेत? ओंकार सोबत
मोहिनी पाटील आणि संतोषी चव्हाण.
3च लोक आहात का तुम्ही?
हो.
बरं, मी प्रेफर केलं होतं 4-4 असले तर ठीक आहे.
बट सर आपल्या क्लासची स्ट्रेंथ 27 आहे ना.
अच्छा, तुझ्या
काही ग्रुपमध्ये 3 असतील, काही मध्ये.
अच्छा. नाही, 4-4चे 7 ग्रुप करा ना. दॅट विल बी बेटर. 7 चोका 28. फक्त एका ग्रुपला 3 येतील. परत 3 करायची गरज नाही. दिस वॉज व्हॉट माय एनिवे. आता तुमचं जर झालं असेल तर आय नॉट मेक मच चेंजेस इन टू इट. नाव व्हॉट यू हॅव डन इज, suppose tomorrow date is Thursday. What is date? 24th.
24.
Okay. Now 24th is a day 0 and the cases are broadcasted. सगळ्यांनी ग्रुपला टाकल्या cases. Everybody has put the case in group. Everybody will able to see the group. What is the benefit of that? So that this case should not be taken by other one. The case you have taken, suppose ओंकारच्या ग्रुपने ज्या केसेस घेतल्या, that cases should not be repeated by other. Therefore we are putting This is the very immediate reason. But
Hello,
next,
sir.
yes,
Uh, Sir, we have already matlab, chosen the topic on which we are going to present the case.
yes, I have
Group.
seen it. Yes. But I'm explaining it in very clear way again. Now,
Okay, sir.
now next thing is what you have put it on group. Everybody will see it. There is another reason why it is put on group, that they will also study the case. They will also go through the case. Suppose the case which has been put by Saili should be referred by everyone, or put by Omkar should be referred by everyone. Okay. But before that, what will simultaneously what is happening there? Now the group of Omkar, they have got 3 people. They will work on the case. They will prepare their own SOAP. Omkar will prepare, then Minakshi will prepare, and who is the third one?
संतोषी.
Santoshi. So Santoshi will prepare. They will prepare their own SOAPs. Now there is a chance that there is a difference in these 3. है ना? ओंकारने जो SOAP बनवला, मीनाक्षीने जो बनवला आणि संतोषीने जो बनवला, there is chance, or rather it is 100% possible that there is a difference. Why there can be difference? First is
Yes.
not looking the things properly. एक चीज बरोबर नहीं बघितली, एकाने एक बघितली, one has seen one type of thing, other has seen other thing. Now what you will do, after you take 2 days for this, 2 or 3 days for this. Now you will 3 will discuss among yourself. Omkar will read your SOAP, Minakshi will read her SOAP, and they will match it. ओंकारने जे लिहिले त्याच्यामध्ये आणि बाकीच्यांमध्ये काय फरक आहे, and they will find out the reason behind it. Now why we are going, I am repeating this, why you are doing this? This indicate, because in case, whenever you are going for any case, each and every line about case is very important. If you miss anything, that there is every chance that you are missing important thing which is important for your case, or its treatment, or its diagnosis, or your assessment planning. So you are checking whether you are missing anything. You will realize that yes, you are missing one or other thing. Second thing is how you are interpreting the things individually. ओंकारने जे interpret केलं तेच संतोषी करते का, तेच मीनाक्षी करते का, you will realize this. Once you make it, then you will discuss among yourself ki why these differences are there and what was the mistake, whether it was the mistake of Omkar or Minakshi or Santoshi or something else. So you will understand that. Similarly, you are going for assessment. Once you go for that, actually major thing, it should come at a planning level. Planning मध्ये काय होईल की treatment whatsoever suggested, whether it is as per guideline, and if it is yes as per guideline, then which guideline, whether NICE guideline, Canadian guideline, US guideline, or Indian guideline, or any other guideline, or from as per the therapeutics book. You will realize this. You have to refer this, and you have to check it which is the most appropriate things to do. I am repeating, I am really unhappy to see that students are not following it, and actually let me tell you, it doesn't make any big impact on me as such, but we are missing very important steps of learning because of that. For me, what you are presenting, how you are presenting is not important that way. It will be important in what sense, that what knowledge you have gained out of it, and all these steps are there to understand that where you commit the mistakes. Understanding where you commit the mistake is also important aspect of gaining the knowledge, which many of you are missing. You are getting thrilled, or you are getting very happy when you just present case in any format, even without proper knowledge, and you feel happy that yes, either you have fooled the teacher or you have done it, you have completed the ritual. My sincere appeal is, you don't go for the completion of ritual. What you do is, you try to get the knowledge out of it. what knowledge you are getting? Suppose I told you 3 people are doing it parallelly, you are matching, then you understand where you commit mistake. Why was the mistake? Whether because you read it insufficiently, whether you interpreted insufficiently, and this is how you develop yourself. What was the problem in your assessment? What was the problem in your planning? This much time nobody can devote, whole class may not able to devote, but you 3 can devote for among yourself. Even if you take around 2 hours, that doesn't make much problem. But once you do it, now lastly you are reaching to a conclusion. When you have discussed among 3, then you feel that this one is the best option. That Omkar has missed this thing, Minakshi has missed this thing, Saraswati has missed this thing, and now when we compile this, then this has become a complete document, and this is the proper guideline, this is the proper diagnosis, this is the proper planning. Now the case is ready with you, and accordingly you distribute among yourself. One will go for discussion of assessment, one will go for discussion of subjective data, one will go for objective, one will go for assessment, one will go for planning, and intermittently accordingly what are the important thing you feel that should be go for point to physician. Particularly when it is diagnosis, it is point to physician. When it is treatment related thing or discharge related thing, then you go for point to patients. Accordingly you distribute among yourself, and that will be the case presentation. Now what about others who have read the case? ओंकारची केस सगळ्यांनी रीड केली, मग बाकीच्यांनी काय करायचं? So they should get questions. If your team has made any mistake, they should try to ask you question that as per guideline this should have been the proper drug, why not this? What was your observation, or why you have not considered this? If you do this, then it is actual learning you are having. Otherwise we can present so many cases, but you will not get the benefit or impact which you should have get. This I have repeated, I think 3rd or 4th time. Now if you have any difficulty, you can ask me. Yes, Omkar or anybody else?
नाही, क्लिअर सर. थँक्यू सो मच. आम्ही आमचं बेस्ट द्यायचं ट्राय करू, आणि आम्हाला जर काही समजलं नाही तर
we are there, we are together, don't worry about it. Okay, only
Yes, sir.
thing is
Thank you.
until and unless you try to give your best, nobody can bring out best from you. तुम्हीच जर अंग चोरायचा, मेहनत कमी करायचा प्रयत्न केला तर तुमच्यातलं बेस्ट कोणीच काढू शकत नाही. त्याच्यामुळे इट्स अप टू यू व्हॉट यू वॉन्ट टू डू, बिकॉज इन लास्ट few years, this is almost 6 year I am observing this. स्टुडंटला त्याच्यामध्ये इंटरेस्ट वाटत नाही, and they just ज्याला we call it as a पाट्या टाकणे, they do it, and unfortunately we cannot do much about it. सो आम्ही जर त्याच्यावर ॲक्शन घ्यायचं ठरवलं तर खूप लोकांना कमी मार्क्स पडतील, अँड दे विल फील की आम्ही तर खूप मस्त पाट्या टाकल्या होत्या, प्रेझेंट केल्या होत्या केस प्रेझेंट करताना सगळ्यात इम्पॉर्टंट गोष्ट सांगतो की डोंट कॉपी वर्ड टू वर्ड फ्रॉम एनी एक्झाम्पल. इट इज नॉट व्हेरी इम्पॉर्टंट टू रीड द मेकॅनिझम ऑफ ॲक्शन ऑफ द ड्रग फ्रॉम द साइट्स. यू शुड बी नोईंग इट. यू हॅव रेड फार्मॅकोलॉजी, यू हॅव रेड थेराप्युटिक्स. तो सपोज आता तुम्हाला 3rd इयरमध्ये एखादा अँटीबायोटिक असेल तर यू नीड नॉट टू गो फॉर गोईंग अ व्हेरी बुकिश reading a very bookish mechanism of action. It is knowing, you are knowing, because in therapeutics my job is not to see whether you understand mechanism of action or not. I can ask it as it is during Viva or something like that. Acha, what is mechanism of action? Just to know whether you know it or not. But my important aspect is whether you know the differentiation of the mild, moderate, severe, which kind of condition it is, whether the treatment which is there that is most appropriate for it or not, most rational for it or not, that is as per guidelines or not, that is very important. So you should give big stress on that, you should have more impact on that. So this is what my advice. I hope you will do it, you can do it in better way, then definitely it will help you, the highest one. Even you can try with this, you just maybe as a experimental basis, you just try that if you do in this way, what benefit you get. Okay, just try this and let me know. Okay.
Sir, tomorrow is Thursday, so tomorrow
already
will be presentation or
already whatsoever cases you have taken, you can go for that and immediately start doing that. That is cases for Monday, cases for Thursday. Tomorrow immediately go for cases for initially you go for cases for next Monday or like that. Again, important thing is
okay.
you specify the area, particularly one area you fix. Area example, the related mostly either related to cancers, related to virals, related to fungal, related, because most of the treatment from immunological you can start with, because it was the first topic which we started with. So immunology, diuretics accordingly. So go for immunology, go for diuretics, then go for.
Sir, according to syllabus sequence, we can proceed with the cases.
Yes, yes, as per syllabus only we have to proceed for the cases. And you may not able to get the cases for genetics or cell related things. You may not able to get any cases in that, so it will be difficult for you, but you will get plenty of cases for chemotherapy, immunotherapy, and
Yes.
diuretics, then hematinics. So you make it, so this will be drugs acting on blood, like that, and you decide accordingly.
Okay, sir.
Is there any difficulty? So it should not happen that one group is giving on hematinics, other is giving on anti-cancer, one is giving on immunosuppressant, one is giving on diuretics. Please don't do this, so that everybody is reading the relevant topic. So our aim is not to complete it or not to have the ritual like this. Our aim is what? That you should get this topic revised. जो टॉपिक तुम्हारे थेरा के प्रैक्टिकल्स को आएंगे उसका रिवीजन यू शुड डू इट. वाइल गेटिंग रिवाइज यू शुड चेक योर कॉन्फिडेंस, तुमने क्या नॉलेज गेन किया है, तुम उसको कितना अप्लाई कर सकते हो, ये तुम्हारा कॉन्फिडेंस चेक करना है. दिस इज़ व्हाट द मोटिव बिहाइंड दिस. तो तुम उसको ईमानदारी से पार्टिसिपेट करोगे तो तुम्हारी लैकुना तुमको ईमानदारी से समझ में आएगा, जो तुमको फ्यूचर में शायद काम में आएगा. और अगर तुमको कुछ नहीं देखना है तो इट्स अप टू यू. ठीक है, दिस इज़ व्हाट माई suggestion. Okay, clear?
Yes, sir.
Okay, then all the best. Fix the time and let me know.
Yes, sir.